Yep, it's time for another episode of Tom Kennedy's Science, and I'm your host, Dr. Tom Kennedy. Now, in today's lecture, we're going to talk about how plants absorb nutrients from the soil. You know, they have to get all of those molecular ions like nitrates and phosphates, and they have to collect all of these electrolytes. Wait, electrolytes? Every time I hear the word electrolytes in plants, all I can think of is Brondo. It's got electrolytes. The thirst mutilator. It's what plants crave. Of course, this is from the movie Idiocracy, where Luke Wilson in today's world was just an average guy, and he got frozen in time for like 500 years, wakes up, and the world is gone, where our world is headed to quite rapidly, actually. And he's like the smartest guy on the planet. And they're trying to water their crops with this green Mountain Dew-like substance called Brondo. Very much like Mountain Dew, right? I love Mountain Dew. Don't get me wrong. And the crops are dying, of course. And he's like, why don't you put water on the crops? And they laugh at him. Water? That's what's in your toilet, man. Because nobody drinks water. So, of course, they water the crops. The crops live and they start to grow. And he's hailed as a national hero because he saved the crops. I know. Yeah, I'm afraid that's where we're headed, to that level of stupidity. But beside that message in the movie Idiocracy that is full of prescience, what's going on here is that the reality is plants need carbon dioxide out of the atmosphere and water from the soil. That's 96% of what a plant is, but they need more. They need, like I said, these molecular ions and electrolytes. And the way they get them, they absorb them through their roots. And in fact, plants have these small root hairs with the sole purpose of increasing surface area so they can mine more nutrients out of the soil. And think about this, a rye plant. These rye plants might have as many as 10 billion root hairs. That's a lot of root hairs. And this creates a zone of depletion around those hairs where they're mining these nutrients out of the soil. And in my last lecture, I talked about those. Nitrates, phosphates, potassium, calcium, magnesium. So the point is how do plants actually uptake these nutrients? So in this lecture, I'm going to focus on these mechanisms of nutrient uptake. So how do we get these electrolytes? Sodium, potassium, calcium, magnesium, and chloride. How do we get those into the plant? And to do so, we have to get them inside the cells in many cases. Now remember this. Go all the way back to your cell and molecular biology. I know, that's a long time ago. And in cell and molecular biology, you remember that all cells are surrounded by a membrane. And that membrane has a property called selective permeability. Now what that means is, it's a fancy way of saying that only certain things can pass through the membrane. So only certain things, it's selective, can pass through, that's your permeability. So small hydrophobic substances, carbon dioxide, oxygen, they freely pass through a membrane. Water, I know, water is hydrophilic, it's a polar molecule, but it can pass through a membrane because it's small. However, your electrolytes and your molecular ions, they are charged. And because of their charge, they do not easily cross the membrane. So the membrane easily excludes your ions, both molecular ions and elemental ions. Now remember, molecular ions are like nitrates and phosphates. They're molecules bound together by chemical bonds, specifically covalent bonds, but they're charged, so they're an ion. And then of course, elemental ions aren't in a molecule. That's your calcium, sodium, potassium, chloride. You get the idea, right? Okay, so plants, they've got to concentrate potassium, phosphates, nitrates, calcium. They got to concentrate them inside their cells. So to do so, they're going to use active transport. They're going to use energy, right? They're going to spend energy And they're going to use some type of transport pump, some protein in the membrane to move these things into the cell and concentrate them. So we're moving them against their concentration gradient. So how do they do that? One way that plants do this is they create electrochemical gradients. Now, this goes back to our cell and molecular biology once again. You know, a common feature of every single cell on this planet is we have a plasma membrane. And every single cell spends lots of energy creating electrochemical gradients. Now, a lot of sources will also call this a membrane potential. But a membrane potential and electrochemical gradient, what they store is potential energy. And what do you need for active transport? An input of energy. Now remember, every type of transport, even diffusion, requires energy. It's just that diffusion and facilitated diffusion, they're using the energy that's already present in the system. Whereas active transport, the cell or the organism, you have to spend energy. One way we can do this by building up this membrane potential, this electrochemical gradient. It's a system out of equilibrium that we can harness for energy, much like storing water behind a dam or how a battery stores energy. Another way that plants can get electrolytes, especially like sodium, potassium, and calcium, is that they can do what is called cation exchange. It can help move those ions into the cell. And we'll talk more about that here in a second. Let's talk about this electrochemical gradient. Now, for those of you that don't know or don't remember what an electrochemical gradient is, there's two parts here, electro and chemical. If I'm pumping something like a proton, which is nothing more than a hydrogen ion, right? Hydrogen is nothing more than a proton and one electron. You lose the electron, what are you left with? A proton. Protons are positively charged. So hydrogen ions and protons are the same thing, right? And they're positively charged. So electrochemical gradient. If I pump protons across a membrane, okay, I create an electrical gradient. And I create an electrical gradient because I am pumping these positively charged protons out of my cell. So the outside of my cell becomes positively charged compared to the inside, which becomes relatively negatively charged, right? So we have an electrical gradient. That means that anything that is positively charged on the outside of the cell will be attracted to the more negative charge on the inside of the cell. Next, a chemical gradient. So we got electrochemical. The chemical gradient is I now have more hydrogen ions, protons, outside my cell. They are out of equilibrium with their environment, right? There's more of them outside than inside. That is also a chemical gradient. So hence electrochemical gradient. And this stores energy as a membrane potential. The reason why we call it a potential is because a potential is the difference between the positive and the negative charge across a membrane. It's the exact same thing as a battery, right? Battery stores potential energy. We measure in volts. So plant cells, guess what? They spend a lot of energy pumping protons. And in this case, to get nutrients, they pump protons outside of the cell, creating an electrochemical gradient. So like I said, if I'm more positive outside the cell and I'm more negative inside the cell, now these are relative terms, that's going to pull your cations into the cell because they're going to go down their electrical gradient, and they might even go against their chemical gradient. It just depends on which one is stronger. If you have a much stronger electrical gradient, you're going to be pulled down that electrical gradient even against your chemical gradient. So you might be able to pull in something like potassium ions into the cell and concentrate them because you're pulling them down an electrical gradient. Now potassium, and I will also say calcium and sodium and magnesium, these are all cations which are positively charged. Now, here's how a cell can start to exclude certain ions. In order for potassium to enter a cell, it has to have at least facilitated transport or active transport. You can actively pump them in, or you can passively allow them in through facilitated transport. But to have facilitated transport, you must have some type of channel protein that allows the potassium ions to enter. And that's one way we can exclude like sodium ions, because yeah, the sodium ions might want to enter into the cell. They can't get through the membrane because the membrane has selective permeability, right? And there's no ion channel dedicated to these sodium ions, or at least it's gated so they can close it. So you can allow potassium ions to enter to the cell through their ion channel, right? That's facilitated diffusion. You have to have some way of letting them through the membrane. Now, how do you get anions in here? Now, two very important anions are these molecular anions, nitrates and phosphates. And in fact, these are some of the most important nutrients to a plant beyond carbon dioxide and water. And in fact, if you buy fertilizer, it's got a number on there, 10, 10, 10, or 10, 5, 0, or whatever those numbers. Those numbers are the percent nitrogen, the percent phosphorus, the percent potassium, because these are three of the macronutrients needed. And nitrates and phosphates are both molecular anions, means they have a negative charge. So the way that a cells concentrate nitrates and phosphates is, once again, they create an electrochemical gradient. Here's our proton pumps, right? I mean, proton pumps in plants are used everywhere. And as a plant is pumping protons out of their roots and into the soil, it's lowering the pH. It's making it more positive, okay? Now, the protons are out of equilibrium with their environment. So what we use is a co-transporter. This is an example of secondary active transport. Here's why. You have the proton pumps pumping protons. The protons, what they want to do is reach equilibrium back inside the cell. So they go through the co-transporter. They're just going down their electrochemical gradient. But as they do so, their potential energy is transformed to kinetic energy. Now we know that we can use energy to do work. And in this case, the co-transporter, the protons, as they go down their concentration gradient back into the cell, they're going to drag the anions with them. And you're going to have a co-transporter for nitrates and a co-transporter for the phosphates. It's pretty cool, isn't it? Now, think about this. Remember we've got the symplastic route and the apoplastic route. Now, symplastic is going through the cells, right? So if we're pulling these anions in, if we're pulling these electrolytes into the cells, and once they get to the endodermis, that will allow those electrolytes and molecular ions, right, our nutrients that we need, through the Casparian strip, through the endodermis, and into the vascular tissue. And now the plant has nitrates, phosphates, calcium, sodium, magnesium, all these things it needs. Well, I said sodium. We'll come back to that. It needs a little bit of sodium, but plants typically exclude sodium. Now, like salt water is mostly sodium chloride. Too much sodium for plants is a problem. And in fact, even our cells exclude sodium. They pump sodium out and potassium in. And if you have too much sodium outside the cell, it lowers the water potential outside the cell. Now, if you lower the water potential outside the cell, where does the water flow? Yeah, it starts flowing out of the cell into the surrounding area. The cell can become dehydrated and you can lose turgor pressure. Now remember, turgor pressure is very important for phloem loading. So not only do your plants begin to wilt because they don't have enough water, but you can now affect their ability to transport sugars and other secondary metabolites, things that plants are making after sugars, right, throughout the plant. So too much sodium is a bad thing. So as you might guess, plants don't like salty soils. So they dehydrate plants and they affect all kinds of things. So the way that plants get around this is they passively exclude sodium. Remember, sodium ions cannot pass the Casparian strip. They don't enter into cells very well. They don't get through the membranes, which are selectively permeable. And there's no real ion channels for them like there are for calcium and potassium for them to go through. So cells and scaling up, roots prevent sodium from entering the vascular tissue, specifically the xylem. However, some sodium does get into cells, and sometimes plants need to uptake water, right? And one way you can uptake water in a dry environment is you lower the water potential inside the cells. And by lowering the water potential inside the cells, water will flow from the soil into the cells, right? And even inside of cells, plants need trigger pressure. They need the vacuoles to swell with water. So vacuoles, they store water, they store minerals. And if you need to get more water into a cell and into a vacuole, what do? You lower the water potential. And one way to do that is by moving sodium. I know I just said that you exclude sodium, but sometimes you need to. You can move that sodium into the vacuole. And vacuoles use antiporters to do this. Now, we just saw a symporter, right, for nitrates. An antiporter, instead of going both ways, protons go this way and sodium goes the opposite way. Hence anti, meaning opposite. So basically, on your vacuole, you basically start pumping protons into the vacuole. Once again, by pumping protons into the vacuole, you create an electrochemical gradient. You are storing potential energy as a membrane potential. We're storing energy like you would in a battery. Now, these protons, they're out of equilibrium with the cytoplasm of the cell. Because remember, we're in the vacuole. So the protons will go through an antiporter. They're just going down their electrochemical gradient. Their potential energy is transformed to kinetic energy. And as it does so, it pulls a sodium ion the opposite way into the vacuole. And of course, once you start lowering water potential inside the vacuole, water will move in there through osmosis. The vacuole will swell, and you'll get an increase in trigger pressure inside the cell, and the plants won't wilt, even when it's dry outside. I'm not going to talk about this too much here, but another way that plants can improve nutrient uptake, especially in poor soils, is to have an association, a symbiotic relationship with mycorrhizal fungi that can actually help improve nutrient uptake in soils. And that is the subject for plant symbiosis, or plants form symbiotic relationships. So of course, I talk about how roots completely mine the soil. Guess what? Welcome to biology. There's exceptions. There's always exceptions in biology. Life finds a way most of the time. I mean, there are constraints on life finding a way. That's one of the lessons from Jurassic Park, right? Life finds a way. There are these plants called epiphytes. Now, epiphyte is a generic term for something growing on top of something else. So in the southeast, where it's a bit wetter than in the southwest. Hey, and for you guys in Europe that are watching me, and throughout, temperate regions and in tropical regions as well, there are lots of epiphytes. And these are plants that are growing on top of other plants. And these epiphytes, their roots kind of grow in the bark. And guess what? In the tropics, they'll actually accumulate an organic layer of soil. And they can mine that as well. And these plants are not parasitic. All right? They're just growing along on the bark. And like I said, they'll even build up an organic layer. And so they can actually live off of in the air. And, in the southeast and in other areas of the world, rainfall for you and I might be pretty constant. A week, you might go 2 or 3 weeks without rain. But if you're an air plant, you're getting really dry. So one of my favorite plant adaptations is the resurrection fern. And it's called a resurrection fern because when it's dry, you can see in the picture to the right, they look almost dead. It rains, and the entire branches of these trees will turn bright green from these resurrection ferns coming back to life. Parasitic plants. There are parasitic plants. They're parasites of other trees. One of my favorites, of course, is mistletoe. One way you can identify mistletoe Let's go out in the wintertime, like around Christmas or Hanukkah. And when you go out, if all the trees have lost their leaves, but you see a dark green patch of leaves up there, that's mistletoe. Mistletoe is a parasitic plant. And they're parasites. Their roots actually grow into the phloem of their host trees. They pull nutrients off of them. And if the parasitic plant, like this particular mistletoe, is green, it can still do photosynthesis. So it's pulling a lot of water and other minerals out. Other parasitic plants are not green. And if they're not green, they're probably not doing photosynthesis. For example, Indian pipes. You know, on the bottom of forest floors, you go out in the woods, it can be really dark. And it can be so dark that there's not enough light for photosynthesis. But some plants have found a way to survive in these environments. They're parasites on the roots of other trees. So what you're seeing is Indian pipes. No pigments. There's no carotenoids. There's no chlorophyll. It's not doing any photosynthesis at all. But it does produce flowers. And it does reproduce. And it gets all the nutrients from the tree that is parasitizing. Kind of cool. There are very nutrient poor areas of the world. And in those areas, you get carnivorous plants. Things like pitcher plants and sundew plants and the Venus flytrap and bladder warts. And you get this in very acidic soils, and you get this in very wet soils as well. And often wet soils go hand in hand with acidic soils, and that makes your nutrient availability very, very, very low. So one of my favorites is the pitcher plant. Those are modified leaves. The leaves hold water, and they actually release digestive enzymes in the water. So when an insect flies in there, they drown. The plant kind of digests the insect, and they get all their nutrients from that. And they're also green as well, so they're doing photosynthesis. And it turns out that there's an entire biotic communities living inside of each one of these pitcher plants. There's also sundew plants. Check out those trichomes. The trichomes are those projections, and they're full of sugars. Sugar's really sticky. Think like jelly, right? So this fly landed on there, and he's like, nope. And the pitcher plant will really just wrap up around it and digest it. There's a floating aquatic plant called a bladderwort. And they're called a bladderwort because they're floating, and in the roots, they've got these tiny little chambers, and they create this negative pressure, and they attract their prey, little daphne and other microcrustaceans in the water. They'll go in there, and then it'll trigger it, and it'll suck them right in, and they'll digest it. And ironically, these bladderworts have some of the most sophisticated plant mechanisms in the world to catch their prey. And then, of course, there's the Venus flytrap. And, you know, the inside looks like kind of like meat. It's got some scent. It attracts flies. The flies trip the hair triggers, and they close really quick. And then they release digestive enzymes. And you know the rest of the story. They digest it as well. Okay. Well, I hope you enjoyed this on how plants acquire nutrients. And yeah, there's a lot more to this story. I didn't go into all of it, but I hope you learned a little bit. All right, until next time.